Okay, thanks everyone for coming in such high number. It's a bit intimidating. So today I'm going to be talking about the work that I did as part of my previous postdoc when I was working with an ACEAS, where I'll talk about the response of coastal Antarctic phytoplankton to changing iron and manganese conditions. That's the first time I'm doing a talk that's this long, so I'm going to use the opportunity to do a bit more of a background and talk about things that I usually don't get to talk about. Then I will present some preliminary results from bioassay that I did during the MISO voyage and the Denman Marine Voyage this year, and then I'll finish with some lab experiments result. So my work focused on phytoplankton. What are phytoplankton? It's these microscopic algae living in the surface ocean. Surface ocean because they rely on light to grow and on the nutrients that are present around them. When you put them under the microscope, they look really beautiful. This is a picture that was taken by Robert on the MISO voyage. It's mostly diatom, which is a specific group of phytoplankton that's very important in the Southern Ocean, and they have this outer shell silicate, and they come in all sorts of different shapes and colors. Yeah, looking very beautiful. Phytoplankton is super important for different reasons. First of all, it's a photosynthesize, so it's hidden a bit by the sign over there. But through this reaction, which is the same as the trees on land, phytoplankton absorb carbon dioxide and release oxygen. And so obviously we care about this reaction from these from the two sides of the reaction, because through this they help inducing this transport of carbon dioxide from the atmosphere into the ocean. While we can find phytoplankton all around the world, they don't grow the same everywhere. So there are some regions of the ocean that are called high nutrient, low chlorophyll region, of which the Southern Ocean is the largest one. As the name describes it, they're characterized by very high concentration in macronutrients. So on the left figure, you can see the concentration of nitrate, which is kind of a primary food source for phytoplankton. And so the color here represents the concentration with orangey red color showing really high concentration. And you can see that all around Antarctica, concentrations are really high. But then if you compare this to this climatology of satellite chlorophyll A concentration, chlorophyll is a pigment that's found in phytoplankton, and it's often used as a proxy for phytoplankton biomass. You can see that despite the fact that we have nitrate all around Antarctica, the amount of, the phytoplankton is not spread equally all around. It's quite patchy. So there's some places where it doesn't grow where we would expect it to grow. And this is because in the Southern Ocean, phytoplankton growth is limited by iron. In this region, there's very low concentration of iron because there's less land, and iron is like a very abundant in crustal material, and so there's just less source bringing iron to the Southern Ocean. So we have a need where we want to understand what parameters are controlling phytoplankton and how it changed spatially, so that we can actually predict what the current changes that are happening, how they may impact phytoplankton growth and how they impact the carbon cycle in the future. There's different ways to study phytoplankton. So as I mentioned, this is a satellite information chlorophyll, chlorophyll A measured by satellite. So satellite have really allowed us to get these really global study, which has been really helpful, but they can only see the top layer of the ocean. And so sometimes they miss the growth of phytoplankton a bit deeper in the water column, which is actually quite common in the Southern Ocean. And then this can only be picked up by actual in situ measurement. So if we visit the area with a ship, or if you have a PGC algo in the area, for example. I don't often get to put pictures of other things than phytoplankton on my screen. So I also wanted to mention that phytoplankton is crucial because they are the base of most marine food webs. And so in the Southern Ocean, they feed the krill, which then have to feed all these charismatic megafauna. And these are all pictures that came from the Denman Marine Voyage. So now that I have them, I thought I could use them. A bit more about the role of iron and manganese within the cell. So both of these metals are essential for phytoplankton growth because they are used in many protein or enzymes, often as cofactors. But these metals also are often used due to their redox state, which I'm not going to describe too much, but they can be used as electron donor or acceptors. within several reactions. If we look a bit more at the photosynthesis, which I'm sure you're all really keen on learning about, this is a highly simplified schematic of the light reaction of photosynthesis. So as its name suggested, that's a reaction that's happening with light. And the goal of this reaction is to produce energy for the remaining part of the photosynthesis, which does not need light. And so in that case, don't worry too much about the different complexes. But if we look at the iron requirement in these, just in this first part of photosynthesis, iron is needed already at several locations, whether it's like at the cytochrome molecule or in photosystem, in different photosystem. In contrast, manganese is only needed for the oxygen evolving complex, but it's a very crucial role. Because it's from this complex that water molecules are split into oxygen. So you could actually say that manganese is responsible for the production of oxygen during photosynthesis. So pretty crucial, important role. Now, if we focus a bit more on this left part of the schematic, so looking at the photosystem 2, whose role is to provide electrons for the rest of the photosynthesis steps. You can look at this schematic that I did on the right. So it's an even oversimplified schematic of this already simplified schematic. And here, what you can see is that there's a direct interaction between light, iron, and manganese within the cell. Because when light hits this complex, it'll be received by the chlorophyll, which will be located on the what I'm going to call the size antenna, the remaining of the talk. And then these photons will be channeled down to photosystem 2, which contains both iron and manganese. And that means that depending on how light tend to photoacclimate, there's a condition where it can impact the iron and potentially manganese requirement of the cells. So, for example, there was a study from Sanda and Huntsman that showed that when under low light condition, phytoplankton will kind of need more of these complexes, so it has more chance to capture the small amount of light that's in this environment, and because of this, it will increase their iron requirements. But since then, there has been more study in the Southern Ocean, including several studies from Robert, that have shown that this is actually not the case in Southern Ocean phytoplankton, because they have a different type of adaptation where they actually have this really large, what I'm going to call size antenna that provide them a bit more chance to capture the photons. So if you, like me, and you tend to overthink a bit why would the phytoplankton include iron and manganese in such crucial processes if they live in a place where there's no actual iron and manganese, like in the Southern Ocean? Well, this is, sorry, spoiler. This is actually going back from the time when life appeared on Earth. I saw that Katerina was here in the room. I don't know if this is supposed to look like, but when I Googled Archean Ocean, which is when microbial life started on Earth, apparently this is an artistic representation that I found, which I thought looked amazing. So anyway, when life started appearing on Earth, which was a very, very long time ago, when I hear Archean, I think about dinosaur, which I think is like a blip compared to when that was, which is why I had the Jurassic Park picture. But anyway, so that was a long time before then. But at that time, the world was quite reduced. And so it means that there were a lot of high metal concentration like iron and manganese. And so when life developed the cell, they kind of incorporated this metal into their processes. That's because it was so available at that time. But then a while later, oxygenic photosynthesis evolved. And so this new organism kind of like slowly oxygenated the Earth. And so this has led to the disappearance, I mean, over a very, very long time scale of like these metals that were very abundant, because when you put iron or manganese in a lot of oxygen, they tend to oxidize, so they form this precipitate and then they fall down the water column. And so we went from a system that was super high in metal towards the system of, toward an oxygenated world, which had a lot less concentration in metal in the environment. And to illustrate this is a result of manganese concentration on a transect from one of my PhD papers. So this is going from Tasmania on the left to Antarctica on the right. Again, the color is showing the concentrations. And here you can see that the blue means that the concentration is really low. And you can see that along this full transect, most of the manganese concentration are extremely low. You can see some increase along the coast, so near Tassie and near Antarctica. And that's because, as I mentioned previously, manganese, like iron, is coming from the crust. And so if you go closer to the continent, there's input of this metal. This is an animation of like the same SR3 voyage. So this is like the 3 transect that we did at the bottom of the line. And you can see that despite the fact that we get more input of manganese from the coast, if you squint, it's a bit hard to see, but if you look at the surface, it's actually still greenish or blue. So despite the fact that we get this input from the land, the concentration still remained quite low at the surface because it's uptake by phytoplankton. So while we've known that iron is limiting phytoplankton growth in the Southern Ocean for many decades, the first experiment that tested this with manganese were quite inconclusive and didn't show any response for phytoplankton growth. However, from a couple of years ago, there has been some observation of mild limitation or co-limitation in different regions. So these are just an example of the main paper that you could find on this type of question. But most of them are studying the Drake Passage because there's a lot of voyages there. There's one study from the Pacific, one study from the Ross Sea, and then 2 studies south of Australia, which are mine from my PhD. Out of these studies, only one is looking at a season that is not spring or summer. Lucky, it's ours. But yeah, which means that, I mean, it makes sense because most voyages are going down in spring or summer, but it means that we kind of have a bit of a gap in the spatial coverage and in the season to know how manganese is controlling phytoplankton growth in the Southern Ocean. In fact, if you type manganese limitation of phytoplankton Southern Ocean in Google Scholar, the first study that pops out is a modeling study that, although being very nice and really interesting, looking at how manganese limitation could become more important in the future. It's a study that used 3 data points in the Southern Ocean to do this whole study and modeling, which is, I think, really stressed the fact that there's a big lack of data for spatial coverage and seasonal gap that the modelers need to be able to make a bit better predictions. So how does typical bioassay work? So I wasn't really sure who was familiar with this type of measurement. So I wanted to go from the start. So usually when we arrive on site and we want to study the response of phytoplankton communities to changing condition, we'll first send the CTD down, and then there's which has all these bottles that will sample water at different depth. But there's also a whole set of sensors on the CTD that gives us information while it travels down the water column. So we'll send it down and we'll get this profile, which will give us an idea of like what's the mixing condition at the surface, what's like the temperature, salinity, what are the features that we would observe. And then if you get this green line on it, it's a measurement of chlorophyll fluorescence, which we use as kind of a proxy to see, okay, it appears that there will be more phytoplankton there. So let's incubate this phytoplankton. If you're lucky, the chlorophyll profile, which is represented by the green dot, matches the fluorescence. And then you're happy with this because then you really sample the feature that you wanted to. Sometimes there's some issue that happens. So this is a case that we had on Denman where we had this, the CTD went down. We had this big chlorophyll chunk from the surface down to 210 meters. And we were like, amazing, we're going to incubate this water to see what happens. And then on the way up, the boat drifted and we sampled completely different water. This happened later in my results, so I'm describing it right now. Once we decided, we have the information from the CTD, and we decided what we want to study. We sample water, trace metal clean water to incubate. So there's always a really big collaboration with the trace metal team. So they put this trace metal rosette in the water, which is basically a CTD, but any metal part that could be replaced has been replaced to avoid contamination with metals. So they put this in the water. Then we have to work on the extra clean condition to not contaminate the sample. And then we collect this trace metal clean water, put it in this polycarbonate bottle like this one, and then change the condition. So for example, I add iron or manganese or a combination. Or you do nothing if you want to control. And then we incubate this community, natural community, under the changing condition in these deck-borne incubators there. So the incubators are kept outside. This is because we want to keep the natural light and night duration for the algae while we incubate them. We can also manipulate the light level using these mesh bags that you can see in the incubators. So by using different thickness, we just decrease or modify the light level. And then, yeah, when you work in Antarctica, it's really hard to grab the bottle because the water is at -1, and it's a picture of Matt, freezing up there. And then after we incubate them for a duration that we decide on the spot, we measure a whole set of like final parameters. So here, I'm going to talk about photophysiology measurement through FRF, but we can also measure chlorophyll, biogenic silica, particulate organic carbon, carbon uptake, or we try to look at the community composition. Anyway, there's a whole set of parameters that we can measure at the end of the incubations. So moving on to some MISO results. So MISO was a voyage that happened in the summer 2024 on the Investigator. You can see on the left schematic the voyage track that I got from Annie, where we went first along the coast and then we went back up to Fremantle. During this voyage, I was part of the biology team, which you can see in the top right corner. We did a lot of sampling from the CTDs to get heaps of info on the biology. We did multiple bioassays, and the really big novelty is that we were running mesocosm experiment, which you can see the mesocosm on the right-hand side. And this was a big collaboration between the biology team and atmospheric team. I'm not going to present any results on the mesocosm, so if you want to hear about it, someone should invite Robert to present a talk. About the incubation that we did. So I'm only going to talk about my iron manganese experiment. These were the different process stations that we did. So a process station is just a station where we stay a bit longer and we do a lot more sampling on the spot to get more information about what's happening. So in that case, we would fill the mesocosm, have multiple CTD casts to get like an uptake cast during the day, know the light condition, then have a lot of parameters on the biology. Initially, we have multiple TMR, they deploy the ISPs. Anyway, we get a lot more initial information, and then we do our incubations. And we had this really interesting feature on MISO while we were near process station 2. We encountered this really high patch of chlorophyll. That was quite interesting too. So I took water from some of these different locations, and then I wanted to know how they would react to changing iron and manganese conditions. So if we look in a bit more detail at the condition when we arrive, so each top panel is showing you the satellite chlorophyll concentration at around the time where I collected water to incubate it. The different red circles show you where I took the water, and if it's colored with an arrow, it shows you that this is the actual station where I took the water. So from process station 1, if you look at the top left corner, I got water from the red circle that's on the right, and you can see that chlorophyll A concentration were really low at that time. Then what I call blob, my sneaky patch experiment, and PS2, were the station that were near actually this chlorophyll patch. So the process station 2 was planned at the end of the transect. And I did another iron manganese incubation when we were actually within this chlorophyll patch. But yeah, it's a bit easier to see it here. And then on the last one, you can see the station, which is in the top left corner. So it was an open ocean station used as a reference compared to the other that were more coastal stations. About the blob experiment. So although, despite the fact that I've had really interesting results with the blob experiment, you're going to see them in a minute, it's quite hard to interpret because we don't know what the initial condition of this was. So when the CTD went down, it was here. And then 2 hours in between measurement, the boat drifted here, and then the TMR locked my bottles here. So I incubated this water for my incubation, but that means that I don't really have initial condition for this specific experiment. And this stress the need that when incubation are happening, drifters are really, really helpful to remain within the same water mass. So about the initial condition at each station, I'm not going to go in too much detail, but these are just kind of a snapshot of the info from the CTD sensors that we had for each station. And the red circle is just showing where we've incubated, where we took the water that we then incubated. I think what I want to stress on this picture, on this slide, is the concentration in metal. So you can see them here. So we had incredibly low iron concentration, 0.03. I mean, the highest that we had was 0.06 at that station, which is incredibly low. Manganese was around 0.1, maximum almost 0.2 here. But yeah, we had like very low iron and manganese concentration initially before this addition. And I think it's also important, I forgot to mention that we deduced from the nutrient concentration that the process 2 station was a post-bloom water. So the bloom that we saw was done and then moved away from this area. So if we look at chlorophyll concentration, which I know is a parameter that a lot of you like a lot. So if we focus on the first process station, we observe stimulation of chlorophyll A production on the iron addition, which suggested potentially iron stimulation. We observed the same one at the blob station with a big increase in chlorophyll. We observed again the same at the PS2, but we saw nothing happening at the open ocean station. Now, if we look more at the photophysiology, this slide is showing you the result of FVFM. So FVFM is a parameter that tells us how well the cells are photosynthesizing under specific conditions. So if we look at this, the exact same principle. Sorry, I forgot to mention that these were my treatments. So if we look at process station 1, we had iron stimulation, but we also had an increase in photochemical efficiency with manganese addition, which suggested that somehow the cell had benefited from a manganese addition. Interestingly, we had no iron effect, and we had a big reduction in FVFM under manganese addition. So that means that when I added manganese, the cell could actually photosynthesize well than when nothing had happened. This is the first time I've ever observed this, and I have not found this in any other paper. So this was a very novel result, sadly, the one for which I don't have the initial one. Then process station 2, we observe an increase in FVFM under iron addition, which is a typical sign that when we added iron, we relieved iron stress. And again, not much happened at the process station 4 because we had no increase of FVFM compared to the control. Now, if we look at the size antenna, so I have a bit of a little reminder schematic on the right-hand side. So this parameter is giving us an idea of how large the antenna appears, which is giving us an information about how connected the antenna is compared to the reaction centers. So a typical answer when communities are iron limited is like usually if the communities are unlimited, the size antenna will be larger, and this will decrease as we relieve this limitation. So this is what we observe here. So when we added manganese and iron, we saw a decrease in the size antenna compared to the control. This result, we didn't see anything with iron alone, but we saw a large increase of size antenna with manganese, which kind of matched the previous slide that said that the algae were really stressed under manganese addition. Interestingly, only the iron manganese addition had a decrease in size antenna compared to the control. So this is a really confusing result, I think. Nothing happened with the PS2 station, which was also quite interesting, and then no significant difference with the PS4. Now, if we want to take it a step deeper, and we want to look at carbon uptake. So this is a work that's done on the ship. So at the end of the incubation, we take a portion of the water and we re-incubate it with radiolabel carbon. And so the phytoplankton will uptake this carbon uptake. And by the end, we'll be able to get a measurement of how much carbon they uptake per day under this measurement. And we use sequential filtration. So we actually stack 3 filters on top of each other, which allow us to separate the different size classes of phytoplankton. So on this plot, I still have my treatments on the x-axis, carbon uptake on the y-axis, and then 3 process station, 1, 2, and 4, the open ocean at the end. And then each line is like a size. So the top line is only small cell, so between 0.2 to 2 microns. The medium one is medium cell from 2 to 20, and the bottom one are the large cell, which were over 20 micrometer. And what we were able to observe with this result was that at process station, when we added iron, we stimulated carbon uptake in the medium and large cells, but not the small one. At PS2, we stimulated all size classes, and we actually had a significant different carbon uptake between the iron manganese and the iron, with higher carbon uptake under iron manganese addition. And at the process station 4, we didn't have any significant difference compared to the control. So as a summary conclusion of the MISO bioassay experiment on this, we've observed sign of iron limitation at the process station 1 and some sign of manganese limitation for the photophysiology. Interestingly, at the chlorophyll patch, and despite the fact that it was our lowest iron concentration, we did not observe sign of iron limitation. And we definitely observed some interaction with manganese, with the fact that when I added manganese, the cell were doing really poorly. PS2 seemed to be iron limited, but we also had a bit of a small sign of iron manganese co-limitation. And nothing happened for the process station 4, which is interesting because everyone tends to tell you that open water of the Southern Ocean are heavily iron limited, but it's not all the time the case. So now I'm going to switch to some of the results from the Denman Marine Voyage. So this was a voyage that happened earlier this year. It was an autumn voyage, and I was part again of the biology team. So this is showing the station that we sampled for bioinfo. We had 2 process stations in that case where we did incubations. So yeah, process 1 here on the eastern side and PS2, which was within the Shackleton Pauline. So what we expected before the voyage, so we went at sea with this climatology of satellite chlorophyll concentration. This is a map that Kay did that showed that over the last couple of decades, in this region, there's usually really low chlorophyll concentration on the eastern side of the study region, but quite high near the around the Shackleton Pauline. This is what we had in real. So when we came across the area, so the red dots are showing places where I did bioassay incubation. And in that case, we had a bit of a higher patch on the eastern side of the region, which was surprising, not what we were expecting. And the Shackleton Pauline had really, really low concentration. So we had a bit of the opposite than what we expected when we came to the Denman study region. This is a map from Katie Naras, who's doing an honor with Delphine and me. And so this is integrated chlorophyll concentration between 0 to 200 meters. And this confirmed the satellite observation that we had actually higher concentration on the eastern side of our study region compared to the Shackleton Pauline, where not much was happening in that case. About the initial condition of the two stations that we had. So the station was really dynamic, so the mixing was a bit complex. But on the eastern side, we decided to take water, I think, from 60 meters. During this incubation, we actually included a light component. So we incubated at 60 meters and then brought them back to higher surface level. This is to stimulate mixing of the water column, and the cell moved back to the surface to see what happens if they have higher light levels. In the Shackleton Pauline, we had a very well-mixed layer from the surface down to 150 meters. But actually, the area was, again, very dynamic. So the background green profile is the station we did the day after. So actually, when we sampled, it looked more like this dashed line, where we had a really strong deep fluorescence maximum, around 130 meters. And so we incubated this at a low light level and brought it back to the surface. And I was wondering why the surface wasn't growing at that location, why the surface community went, growing more. So I just incubated this too. About some chlorophyll result. So this is the same principle as before by the end of the experiment. The light represents the light level. So obviously the clear bar plot are the highlight level, and the dark one are kept at low light. So in that case, not much was happening when we incubated the communities at light level of 60 meters. But when we brought these communities up to level of I think 20 or 30 meters, it's suggested that they needed iron to be able to produce more chlorophyll. And if we look at this plot, so in that case, we are still on the highlight. So the top one that was included at highlight, and then the blue one is what I call low highlight, so the one from the deep that was brought back to a higher light level. But we didn't have any significant result, significant difference, although we can see that there's a trend in increase in chlorophyll with the iron addition. If we look at FVFM, which just reminding you is how well the cell are photosynthesizing, we didn't have much significant result again, but a trend with like an increase in FVFM with iron addition for the PS1. And we had a significant result on this for PS2, where the deep communities that were brought back to the surface needed iron to be able to photosynthesize better. And not much happened with the size antenna. The only significant result that I got was from an iron manganese addition, actually, but it's just because my error bar are quite wide. So as a summary, what you've seen with this is that despite the fact that we expected higher concentration on the shelf at both seasons, we did observe some signs of iron limitation, which suggests that communities even near this high concentration could be quite often iron limited. But we still have a lot of data to analyze, so this is going to come here in the coming weeks and months. Still going to be focusing on this. And now I wanted to talk a bit about the bioassay in general. So bioassay are amazing to give us a bit of an indication about what's happening at that time in this specific condition, what is controlling the phytoplankton growth at that location. But it's a community response because we don't really know on the spot who's there, and it's actually really hard to constrain properly the community composition in the field. You have to do a lot of measurements, and we're often limited with the amount of water that we can collect. And if you remember this slide of my carbon uptake, so this was from MISO, process station 1, you could see that when we separated the different size classes, we were actually able to see that there were a difference between the small size class compared to the other size classes. So these two were iron limited, well, appeared iron limited, while the top one didn't have any change with iron addition. But if you look at the bulk answer from chlorophyll measurement, you can see that the signal of the large cell is just drowning everything else, right? So by just measuring bulk measurement, it's really hard to disentangle at a more refined scale what's happening actually for the different phytoplankton group. And the best way to do this is to do laboratory experiment. So laboratory experiment allow us to kind of like look at a bit more refined answer and to really study the species niche. So how are they reacting to this this wide gradient in light condition, in temperature, or in these different concentrations? So here I'm just going to present really quickly some result that we did on a laboratory experiment with Robert a couple of years back, where we worked on these 4 different species. So these are 4 Southern Ocean diatoms. The top left one is one associated with ice environment. The top right one, Proboscia, is a typical diatom of open ocean waters. And then the 2 small bottom one were isolated from one of our experiments at the SOTS station. If you're lucky, when you isolate some species, you might get a new species out of it. So this is actually a new species that was identified as part of the Fragileiopsis genus through genetic analysis, and it's actually the smallest of its kind. So the maximum size was 2 to 3 microns, so really minuscule. And we are in the process of submitting the paper to describe this new species, and we're trying to call her Fragileiopsis yetii, which is after Robert's cats. And when we culture the algae, we use this type of tube. This was just so that you would visualize with like a light that we control the level. And then you can change all the parameters that you want. So I'm not going to go in details with these results, but this is just an example of a normalized growth rate from these 4 different species under a range of different treatments. So if we take this a bit in a simpler way, take the color as like the type of limitation that I was intending to have. So blue, I was trying to iron limit the algae. Green, I was trying to manganese limit. And yellow, I was trying to co-limit them. With the purple one being the replete treatment, where they had everything that they could need to compare it to. And the more you move towards the right of the color, the more limiting the treatments become. And I think when there's a skull, obviously means that the algae didn't manage to survive this treatment. And what we saw already from these different treatments was that despite the fact that some of these algae were coming from close location, they had a really wide range of their iron and manganese requirements, which stresses that really different species won't be able to withstand the same state of limitation, and so that it's really hard to get this from an actual community bulk response. If we look a bit more in a bit more detail at the photophysiology, so for the last two species, so this was the southern, the open ocean one, and this is the one that was associated with sea ice environment. We measured some photophysiology. So again, FVFM, how well they photosynthesize, and then the size antenna downstairs. And then just to highlight two really interesting results that we had. Go with the stick. So this is my most limiting manganese treatment. And Proboscia didn't do really well under it. So when I removed all the manganese from the treatment, it was very hard for her to photosynthesize properly. But then keep this incredibly low manganese concentration and then remove all the iron also in the media. And suddenly she was photosynthesizing a lot better than when it was only manganese limited. And this suggests that maybe there had been some interaction between iron and manganese and competition to go within the cell. And another interesting result, so for the size antenna, so as I described before, you can see it really well if you look at these two blue plots here. So usually when southern ocean species are iron limited, you'll see the FVFM decrease because they'll photosynthesize less well, and you'll see the size antenna increase. And so we could see this really well with that species. And so we were wondering if that was going to be the same under manganese limitations. And for that species, it was, because you can see that when we limited manganese, the FVFM decreased and the size antenna increased. However, for Proboscia, we didn't observe this result. So this is the size antenna of the replete, which is exactly the same as the size antenna of my most limiting treatment, which means that depending on which species you're using, it might be possible to identify iron and to differentiate manganese limitation in the field if you have this type of measurement, depending on which algae are actually dominating the community. So we did this test on 4 species, and 3 out of the 4 species showed the same result as Proboscia, which would suggest that most species maybe will have this different photophysiological response. This is a work that I'm doing as part of a Primo mentorship program. So Primo stands for Physiology and Rates in Microbial Oceanography. It's a SCORE working group. And we are trying to build a model up as part of this mentorship to look at iron and manganese within the photosystem too. So in conclusion, you've seen that iron distribution doesn't always mean iron limitation, and that sometimes the limitation doesn't really happen where you expect it to be. You've seen that different species can have a very different iron and manganese requirement, and that it's really hard to capture this from like bulk community response. And I think this really stressed the need that we need more understanding about what's happening. So more lab and more field studies with more coverage and more seasonal, filling the seasonal gap if we want to provide more robust data so that modelers can use this to predict a bit better what could happen in the future. And I would like to thank all the people that made all this work possible. Thank you.
Thank you, Pauline. Are there any questions?
Thanks, Klaus. Thanks, Pauline. So it's really difficult to pull apart some of the overarching lessons because it is complicated. And I was wondering whether some of that complication can come from maybe in your first results where you sampled in the bloom and then outside the bloom. Yep. Could some of that be explained by the fact that some of these phytoplankton communities have already done some luxury iron uptake? They already have really high iron in their cells. Yeah. And as you add the iron to the medium, they're not necessarily needing that. And the other comment was I was wondering, are your, when in the second part, were your isolates, were they azenic or I don't know how to pronounce that, axenic or azenic? Were they by themselves?
No, this are some bacteria.
Okay, so they're bacteria. Yes, because I was also wondering about competition for iron with bacteria.
Yeah. Yeah. Thank you. Yeah, I was told this one doesn't work very well. Yeah, so I think we were trying to think about this result about why there's no iron limitation in the blob, and we were thinking that's probably there had been a spike in this water before, and they were still comfortable with that iron. So I think the question is, how much longer can they stay with this luxurious? With this iron reserve, and we see it when we try to limit the algae on the lab culturing experiment because we need many transfer before they actually start. Their growth actually starts slowing down. So I think that's definitely could happen in the blob experiment. And the other what was the other question again? Yes. So when I did this experiment, it was at the end of my PhD, and we decided not to kill the bacteria from the media because apparently it can be quite complicated to really remove all this, and sometimes you can lose the phytoplankton. And because I did really a lot of treatment, I think we didn't want to add some complexity to it. But yeah, so there's definitely this to take into account.
Does that potentially explain the counterintuitive result of the manganese? So when you remove the manganese in that last experiment,
yeah,
you see that the photosynthetic efficiency decrease.
Yeah.
And then take away the iron next. And then suddenly does that then cause then the bacteria are then competing for the iron as much because you've actually removed the competition. Bacteria, I don't know, like that's just me.
Yeah, no, actually, I haven't thought about this, but I actually have flow cytometry data, so I have bacterial count for this data, so I could look into this in a bit more detail. I think what I was thinking about for this was that if my manganese treatment, the lowest one that is here, that one, there's actually the iron is completely replete, so there's really a lot of iron, and so I thought that this difficulty was coming from like probably the transporter within the cell bringing more iron when they should be bringing manganese, and so the cell's struggling at that level. But this is still a work in progress, so still working on it. Yeah.
I think there was a question. Ilaria, do you want to ask your question?
Oh, do you mean you want me to just like, yeah. So I was wondering whether we understood the sources of iron and manganese around the Denman region.
I didn't understand the first part of the question. What was it?
No, like just if we figured out where the iron and manganese sources were during the Denman campaign.
Well, the trace mineral data haven't been analyzed yet, so you have to wait for the numbers. Yeah.
Okay. Thanks.
Any other? Oh.
Thanks, Pauline. Some amazing data there. Given the sort of striking differences in the lab treatment between the species, what do you think could be driving that, given that at the start of the talk you showed, in terms of the photosystem, you sort of showed the sort of stoichiometry of where iron's needed and where manganese needed. Do you think there can be some variation between the species, or is it pretty hardwired in? You need to have 4 manganese for oxygen evolution, for example?
Oh, there is one. Okay.
Oh, that one. Just in front of me. Sorry. Can you hear me? Yeah. So I didn't mention, but I said that iron and manganese are both used in different enzymes. So there's also manganese is used quite a bit for the defense against reactive oxygen species. And depending on the species, they'll be using one that use manganese, one that use iron, one that use copper, one that use a mix of all of them. So I think like this could be a reason to justify the difference. But I think all of them use 4 atoms of manganese for this oxygen evolving complex. But I think it'll also depend on their strategies and if they're already iron limited. So I think there's definitely, the other molecules that I used And yeah, I think some more stuff to look into.
Thanks, Pauline. That was great. I learned so much. I had a question about this open ocean sample you had from the MISO voyage, I think, and that it was showing it wasn't iron limited, I think. And if I recall from the start, you're saying most of the studies have previously been done in summer, and this study was in autumn. I was just wondering, could it be a seasonal signal you're picking up, or do you think it's just a...
Were you asking about this results?
Yeah,
specifically?
and the PS4
So this is
one.
actually, it's still summer. So the MISO was a summer
Oh,
voyage, and
okay,
the Denman was an autumn voyage.
Got it.
Yep.
Sorry.
Oh, that's okay.
Another question in the room or online?
Hi, Pauline. That was a fantastic talk. If you just go back to the slide that you were at before, showing the difference in the incubations and the FVFM and the antenna size, I was wondering, you mentioned between the two species, you might be able to identify in the field
Oh, yeah. Yes.
something.
Yeah.
And I was just wondering what the advantage is to that and what you might be able to do differently in the field if you can identify these different processes or these different species.
Okay. So I think the good thing for this is like if we had something in a theoretical magical world automated that could measure this dark acclimated VFM and sigma, then we'd be able to get information about iron limitation and potentially manganese limitation in real time, right? But I don't think this exists yet. So yeah, the fact that 3 out of the 4 species that we tested had this different answer when the size antenna didn't vary under manganese limitation, but it did under iron limitation, means that potentially if we were finding this result, it could be an indication for this. But this is, I think there's a need for a lot more studies to constrain this because this will change depending on the species, the condition, the light condition and things. So it's still quite fairly, it was like an interesting result, but it's still very at the preliminary state. Is definitely not ready to go and identify in the field. But I think it could have cool application if this is something that we can reach a bit later.
Thank you.
Any other questions from the room or online? No? Well, then I think thank you very much, Pauline. Put our hands together.